This recording is for the fall semester of Bio 282, the Microbiology Lab. This is talking about scientific communication, about posters, the poster session, what that looks like. I'm also going to go through the Tiny Earth Figures assignment and the poster assignment. This is the Tiny Earth Figures assignment. It is a writing assignment. It's due Sunday, December 12th by midnight. This can be resubmitted as it is a writing assignment. So the goal is to continue building your experience interpreting results and communicating your findings. This also expands on your knowledge of antimicrobials and antimicrobial producing microbes. The most important part of a research article or poster presentation is how the data is presented in graphs and images and sometimes tables. So for this assignment, you're going to make several figures that you may or may not use on your poster at the end of the semester. You should have 2 figures displaying your antibiotic production of your soil isolates in 2 different ways. One must be a picture of your antibiotic production assay plate, and the other should be some sort of graphical representation of the data. It can be creative. I'll go into that in a minute. And they should be clearly labeled. The key grading procedures here are going to be on APA formatting. They should include a descriptive figure legend, and the figures you submit for the assignment do not have to be the ones you use on the poster. So the first figure must be a picture of your antibiotic production assay plate. So this is when you spread plated the escape relative and then you patched on your microbes. It's that results day. So you must have a picture as one of your figures for this assignment. So you want to follow the APA formatting for figures. I've linked the OWL-Per2 website right here. You must include a figure number. It should be on its own line above the image. Should say figure 1. All figures and tables in the paper are numbered. Anything that's not a table is a figure. So I don't want to see image or any other word other than figure or table. So this should be a figure because this is a figure assignment. Your title should be descriptive and it should be italicized on its own line below the figure number. It should clearly state the conclusion shown in the image. Call out which antibiotic producers there are. And then below the image, there should be a short paragraph describing which isolates had clearing, indicating antibiotic production, so that your viewer can read that. Your next figure can be a graph, it can be a picture, it can be a pie chart, it can be a Venn diagram, it can be some sort of clip art or graphical explanation of the underlying process. You can be creative. I will give you an example of this. This should be formatted the same. It should also be figure 2, then a title, then the image, then a description below it. Figure should be bolded, title should be italicized, each on their own line, and then the note below can be italicized. Here are some examples of Tiny Earth figures. So you note the figure 1 and the figure 2, and then they have an italicized title. This one has an italicized title. Note that the chart itself has a title, but this is in the writing in your Word document. It has a title with the figure. And then you have both images or chart. You have your note below that calls out inhibition is shown for isolates 1, 2, 7, and 12, that it's very clear what those results are. You could even add like arrows to your figure to help call out some of that data if you wanted to emphasize it. Over here, we have labeled axes. So this one is the number of isolates producing inhibition. So we had 4 isolates that produced inhibition against B. subtilis, which is that plate you see to the left. Of those same isolates, 2 of them produced inhibition against E. coli, 1 against P. putida, and none against E. raffinosis. And so then we also note how many isolates total were screened, and that isolate number 7 had broad spectrum use. So it was effective against multiple strains. This is another example that one of my students came up with that I think shows really well all of those interactions, and it's fun. So this is a Venn diagram. You can see that they tested their microbes against B. subtilis, S. epidermidis, P. carnivororum, and E. coli. There were several that were only effective against one, so that was like number 12, number 14, number 3, and number 4. Number 7 was effective against 2 species, S. epidermidis and P. carnivororum. And then number 10 was effective against all 3 of them. So you can see the very specific overlaps there, which was kind of cool. So this was done very well. When talking about scientific conferences, you will have oral presentations and then poster presentations. We already talked a little bit about oral presentations before your unknown microbe project. Lots is happening at the same time with oral presentations. There will be timed sessions. There's often multiple rooms. And it's really important to stay on time and to schedule so that people can shift room to room to see the different presentations they wanted to see. Poster presentations, however, there's a lot of people milling. It's a lot more informal of a presentation. So an oral presentation, you have the stage and you are entertaining the audience with a PowerPoint, versus a poster presentation, as you can see in this picture, it's just a lot of people talking and having conversations. So it's a lot less stressful, in my opinion. So every spring, Thinking Matters happens in the Abramson Center. This is a picture from our Thinking Matters from one year. I don't recall which year. It is all USM and SMCC student research that's happening here. So those are all local presentations. This is an example of what a conference schedule might look like. So this is for the Northeast National History Conference. It typically happens in Burlington, Vermont. You see at the very beginning, they've got a registration table opens. There's setup time, and the exhibition's open, and then overlapping that time, you have your PowerPoint previews and you have your setup for your scientific posters. So that's all that setup information. And then at 8 a.m., the oral presentation starts. So there's going to be a session at 8 a.m. where you can see the schedule for all of those different rooms and those different sessions. That ends around 9 and 9:10 a.m. until about 10:10 a.m. So for that next hour, there's the morning poster session. So half of them will be there and they will present their posters. And there's typically beverages and light snacks and people milling around the room. This is the first of 2 presentations for the day. So all posters remain up for both sessions in this conference. Ours will not, but we will simulate this 2-session thing. So then they have an afternoon session that happens in between some oral presentations, and it just gives people a chance to break and talk and learn about other students' presentations and research. And then at the end of the day, they take down their posters. So the presenters are only required to be present for the time that they're scheduled for their presentation. They're not required, if they presented in the AM session, they don't have to be there for the afternoon session. They do have to pick up their poster at the end of the day, but they don't have to stand next to their poster during the other session. This is a poster that was actually presented at our Thinking Matters. This is a template that Thinking Matters provides. This is a perfectly acceptable poster. This is the way posters have been done for years and years and years. I dislike this poster format, but I will accept it for full credit if you are using it. This poster, there's nothing wrong with it, as it is, I actually don't even know the research. This is something engineering, I believe, about a computer chip, which is cool. You see that they have their abstract in the upper left-hand corner. Remember when we wrote our abstract, that is a short 1-paragraph summary of everything that you did in your research. Then they have their introduction So that's all that background information of like, hey, this is the stuff you need to know in order to understand this and why we're doing it. Then there's the objective where they really highlight, hey, this is why we're doing it, your purpose, your goal, your hypothesis. That's right here. Then they have some bulleted methods. So they're going to tell you how they tested these things, what they did. You've got figures in the center here that are showing you their research and their data. They do have labels on each of them. Then they have their results. So this is part of your results, the center panel. And then you have your results here where they write out what they found, just like we did in our papers. And then they have their discussion, conclusions, and next steps. So this section is where they talk about why this matters. This is often bullet pointed. This one is. And then they would tell us what they might do next if they had more time, more money, more everything. Or if somebody were to pick up the research after them, where could they go with it? And then they'll have a small acknowledgement section and a references section. So this is very traditional. I think we can do better. This is a poster from a student that studied in Kat Miller's lab. I very much respect this person. If you look at this poster, you probably cannot stare at it for 30 seconds and tell me what they did or what they found. There's too many words. It does have a large focus on their results here, which is lovely. But there's too many words. It's too blocky. There's just too much included. This is another poster. It happens to be made by me. I think that it does a good job of focusing on that research and getting away from so many words, but it's honestly still too wordy. Like, it could be better. My title could be more descriptive. I could have actually called out the main point of what did I find in my title. You'll note that the only paragraph of text I have is up here in this background. So it gives you the information of why we did this research. And then you'll note it's numbered so that you can see where the research data is and sort of follow it around the page. And then we talk about number 2, and there's it graphed. This is a visual graphical representation of our methods. You notice there aren't any words on that, and I would just explain that as I'm standing in front of them. Here is my acknowledgments. There are some figures that have information. My conclusions. I had IACUC approval, and I put that down here. So this could be better, but we're moving in the right direction. This is another poster that is moving in the right direction. This is actually by our graduate assistant, Anna. So this talks about her research. And so it gives you the background. It highlights that hypothesis. It calls it out in its own section. It has their methodology. This is a pilot study. So most of their research was actually focused on figuring out the methods. That's why this is so large for them. So it has lots of graphical information here. It calls out the key points and bullet points. It's got bolded sections to help draw the eye. It's got their results in a box. They've got arrows kind of directing you around the page. It's got future plans. It's got sort of all of the same things, but it's organized better than those first 2 posters. Here is a poster not from USM. This is from the University of Bristol. This was done after the Better poster. So we're going to talk about the Better poster here in a little bit. This was submitted as one work from the Better poster template. So this is their whole poster, and then I'm going to dive into it in a second on the right-hand side. You'll notice they have a huge, this is what they did. You know from looking at it and glancing at it, they're searching for new antibiotics in deep sea sponge bacteria. You probably will remember that. They've also got a catchy little cute icon. This one is pop culture, obviously. So you've got SpongeBob, and he's searching for something. So this is very, helps your brain connect those 2 points. Then you have their introduction. Their introduction is bulleted. It's large enough that you can see it from a distance. They have bolded terms so that your eye is drawn to the things that you need to know about. Their methods section is graphical, so it's just images with a title. You don't need a description of each thing. You just need to tell us what you did. And that way you're standing in front of your poster and you are able to give these descriptions. They have their results are very, very defined, also bulleted, also using bold to call out the important things, and tables and graphs. They've got images here of their actual research, and again in their future work they call out important things in bold. So this is really well done. The other thing is that they've got a QR code down here where you can take a picture to download the poster or even get more information is often what's done there. So, quote from Mike Morrison. This is the Better Science poster creator. So this is a researcher who is trying to make better posters. Design isn't about making things look pretty. It's about directing their attention. Great design is subtle. And when you're designing something, it should feel like problem solving. User experience designers feel a surge of happiness when they get to delete something. When it looks too simple to represent the amount of time, the number of hours that you put into it, then you're done. So what he's saying is that you write everything and it looks like one of those big jumbled posters with all the words, and then spend your time paring it down and making it very, very simple. When it looks super simple, then you're done. But it still needs to convey those points. Here is the USM Thinking Matters template. If you would like to use it, you can. This PowerPoint will be posted on Brightspace, so you can download it and just copy that slide. This is obviously on YouTube at the moment, but this poster and whatnot will be on Brightspace. Here are some examples from the Better Poster research. So this is from his first edition of the Better Poster. His idea is that the main finding goes in the center. You talk about it in simple words, plain English, emphasize what you did, and give something really big talking about your point, your graph, something that helps them understand the main points. You also include a QR code so that they can get the full paper that you wrote. We are not writing a full paper for our posters, but typically you would. You would have all of your traditional information, your title, your presenter, your affiliations like USM, might go up there, might come down here. Tiny Earth you could include. There's all sorts of stuff. They have the background, methods, results. They'd have discussions. So this is your traditional bullet point and do your writing on this side. He calls this the silent presenter column. So when they're walking by, they could read this, read your main finding, and understand what was going on in your research. Whereas you would be standing on this side of your poster, and this ammo bar is anything that you feel that you need extra information, data, graphs, tables, whatever you need. So you'd sit on this side, you'd be standing, you'd explain your research, and if they ask you questions, you could point to your extra ammo bar research and be like, hey, yeah, the main point is that we found this, but yeah, this is actually some of the other stuff we did. Another version, they did the Better Poster 2, so he has since revised it based on current research and how people learn. So the hero figure has that sort of left-hand side, still has your background, methods, results, and wordiness, but it's really small. Then you have the main point up in the top. It has that main call out. It has some sort of figure, and the color is important choice to what you're doing. So they're talking about bad mood and angry. Those feelings are evoked from red. They've got this little clip art of a woman who's clearly swearing here. So this helps, is like the SpongeBob. This helps you remember when people are in a bad mood, and they bolded it. At work, it distracts them. They bolded it, which hurts their performance. So this is the thing that you're going to learn from this poster. So they have their figures down here and they can explain it. And then this is all of their writing. So this is the hero figure. This is another example of the hero figure. So you teach people something cool you learned in 5 seconds as they walked by. You've got your little graphic. You've got your major chart information here. And then you've got all of your background info sort of small on the side. Notice they also have the QR code and sort of smaller things here, allowing you to get more information. This is called the presenter. So you have sort of that same call out, the main point that you're picture this, and then you've got some sort of graphic that helps you describe that on this side. And then on the right-hand side, you can think of each of these as like their own PowerPoint slide that you printed and put on the poster. So this is like 4 different PowerPoint slides. So they call it the presenter, as if you had 4 PowerPoint slides to present with. This is another example of the presenter without the like same words. So teach them something cool with your illustration. They've got their method in the top one. Then they've got 2 other ones here. So instead of 4, they use 3. Same concept. Any of these templates are great. You can create your own template, so you can use and copy any of these things. This is your Tiny Earth scientific poster assignment. Your first draft or outline of your poster is going to be due in class with a check-in with either myself or Anna. You must check in with us and get approval that you are going in the right direction and meeting expectations, as our poster session will be open to the public. We want to make sure that everybody is confident and nobody is embarrassed if they are not on the right track. The last chance for you to do so is going to be December 8th for sections 1 and 2, or December 9th for sections 3 and 4. This will be done in class. It is a you did it or you didn't in order to get the points. Your poster presentation itself is due to Brightspace on December 9th for sections 1 and 2, and December 10th for sections 3 and 4. That's 11:59 p.m. is when those are due. You will be presenting in class the very next day. The way our poster session is going to go is that it's going to happen during your regularly scheduled class time, but they're going to be grouped into 2 sessions of approximately 45 minutes each. You're going to be assigned to present at 1 session during your class period. You are expected and required to attend at least 1 other session as an attendee. This can be accomplished entirely during your regularly scheduled class period, but you're welcome and encouraged to attend any of the other sessions. So for example, on Wednesday, our class goes from 8:30 to 10:20. So there are 2 sessions that happen during that class. We'll divide the class in half. Half of you will present during session 1. The other half will present during session 2. The people that are presenting during session 1 can be attendees to session 2, or they can attend any of the other sessions. The people presenting in session 2 can attend section 1, or they can attend any of the other sections. These are all happening during your regularly scheduled class. So the goal of this assignment is to show your understanding of experimental design, your ability to interpret results, and to communicate scientific data. What is a scientific poster session? So scientific poster is a way to present research findings and is typically presented at a conference or symposium. There's 2 parts: the physical poster and presenting it. You're discussing with conference attendees, including researchers, students, experts in the field. And then the physical poster displays the background of the research, the purpose, findings, implications. And the presentation portion is typically just the researcher standing next to their poster and discussing their research with attendees. This is a great one-on-one time to have small conversations and really get into what you did for your research. So this is more of a discussion than it is a presentation. However, the researcher is expected to be able to explain the key aspects of their research, including the research question or purpose or hypothesis. So that is all the same. You don't need to have one of each, but you are expected to be able to tell us why you did this and what you thought was going to happen. You are expected to be able to explain your methods, your key findings, how you analyzed your findings and figured it out, what are the implications of those results, and then talk about the potential next steps. What would you do if you had more time? You, as the researchers, will engage with attendees, answer questions, and explain your research in a clear and accessible way. So as far as the content, again, you're free to utilize the Thinking Matters poster template, any of the better poster designs, or your own design. Make sure you start with the question, if I could only put one piece of information from my research, what would I want to tell the audience? So think about that question. That should be reflected in your title. Each poster style should be able to have that key piece of information as the highlight, the focus, the center of your attention. You need to remember to include all of the other topics, but make it easy for a person to walk by and gather that key point that they need to know. Remember to utilize your sidebars, small text boxes, QR code links to fill in the rest of the required information. Your text should all be short, concise bullet points. It does not need to be flowery or extra wordy. I encourage you to take a moment, walk through the building, and look at posters that are actually hanging in the hallways. Analyze them as a viewer. Do you understand what the purpose was? What they did? What they found? Is the poster visually appealing? What inspiration can you take on how to design your poster well, and what would you do differently from them? There are a lot of cool posters on the 3rd floor, right underneath our lab in this wing. There's lots of posters throughout the entire building. So please feel free to walk all of the floors and all of the wings. All right, you need to cover the following topics. I had them in bold on the other page, but here is them in detail. This is the research question, hypothesis, relevant background. You need to highlight it. Your main objective, your purpose, your hypothesis, research question of your work. You should all know what the purpose of the Tiny Earth research project is at this point. So you should be able to explain what the purpose of doing this research was. You should provide enough background information for why we should care about your research question. Remember that day we looked up all of the antibiotic resistance, all of your soil crisis, all of your antibiotic production, the escapes. Then you wrote an introduction, or you didn't write an introduction, but you could have written an introduction for this section. What would you put there that would tell the person, hey, this is what's going on and what you need to know in order to understand why we did this research? Your methods section should outline the experimental approach you used. So this should either be bulleted or a graphic flowchart of the methods. This should include things like soil collection, your serial dilution, your assay plate, your library plates, those sorts of things. It does not have to include only those. It can include more, but you should include those ones. Your key findings. So present the main findings of your research. So this may include data on the number or diversity of antibiotic-producing microorganisms isolated, may include results about antibiotic screening tests, talking about your zones of inhibition, your microbial susceptibility. It may include information about the microbes you sequenced. Are they already known antibiotic producers? Make sure you use visual aids in this section, graphs, tables, images of your plates to help convey your results effectively. You can use images that you made for your Tiny Earth Figures assignment. You do not have to use those figures. You can make different ones, but you do not have to do extra work. You can use the ones you've already made. Be sure to focus on the key findings and trends. Present the most relevant data to support your conclusions. The interesting thing about creating a poster, you do not have to include everything you did in your key findings. What's the important point you're trying to make? I probably am not including my soil dilution plates here. If my point is this one microbe produces an antibiotic against this one bacterium, I'm going to include that in my methods. That's how I found it, but it wouldn't be highlighted in my findings. It wouldn't be a major thing. If you have really great results with your chemical extract, cool, maybe that's highlighted here. Maybe you didn't. Maybe your sequencing worked and you found a really cool microbe. Maybe your sequencing worked and it wasn't such a cool microbe. Like, you do not have to include every piece of data that we did, but include the most important things. This is where you are determining what you want to present. Implications and discussion. Interpret your results. Discuss their implications for antibiotic discovery. Consider the potential applications of your findings in combating antibiotic resistance. Discuss any limitations, for example, small sample size, potential contamination might have happened, need of more refined methods, replication of results. There are all sorts of things that you could talk about that are errors or that could limit your study. Were the results what you expected from your sample site? Did you get something from what you thought was really rich soil and was going to have tons of results and you didn't have anything? Or did you get lots of results and you just thought, like, this was my backyard, I didn't expect it? What factors might play into the success of one site over another? And then what are the next steps? Explain what you would do next with your current samples if you had more time, all the money in the world, and an interest in this, whether or not you are currently interested. And then suggest next steps for future research. So explain that. So all of these things do not have to be words on your document. Highlight them. So this should be one sentence. Your methods might be graphical, might be pictures, just like mine was. Your key findings, this should be the majority of your poster. Your implications, discussions, and future research, this might be some bullet points surrounding your key findings. That's how you include all of this information together. Your presentation guidelines. So that was how you make your poster. That's your content. This is how you present. So most attendees will start by asking something to the effect of, tell me about your research. What did you do? So then you should have some short elevator pitch walkthrough of your poster. You should be able to point at your poster, explain through all of those different sections, and tell them what you did. Make sure you tell them why you did it, how you did it, what you found, what that means for antibiotic discovery, and then where you might go next. So it shouldn't take you long. That should be like 2 to 3 minutes. You don't have to read every point on your poster during this bit. Give them a second to you want to give them an overview. And let them have the chance to ask you more questions. Then attendees will either say something to the effect of, that's really cool, good job, and they walk away. And you're like, oh, all right. Or they'll ask more questions. Some possible topics people might ask questions on. Can you tell me more about your methods? How did you figure out if a microbe was producing an antibiotic? I don't understand what PCR is or what gelioelectrophoresis is. Why did you sample that location? What was the rationale behind that? What was your favorite part of the research? Do you know if the antibiotic is new or has it been discovered? Is that a species of bacteria known for producing antibiotics? These are all things that, they just kind of lead in. They might continue asking these questions. It may not be all of them. I don't know what questions you're all going to get asked or you're going to ask each other. I would say be nice to each other because you want your colleagues to be nice to you when asking questions. So I wouldn't throw any trick questions out there on anybody. Your submission of your poster to Brightspace should be a PowerPoint slide, a Google slide, a PDF, a PNG, or a JPEG. This is a singular PowerPoint slide. This is not multiple slides. This is one. If you are looking to use another format, please talk to me about it in advance. Then we also have your attendee participation. So this is how your grade is broken down. So you have your poster. This is your content. So your Tiny Earth poster is worth 50 out of those 100 points. 10 of those points is you checking in with me or Anna and getting the approval that you're going in the right direction. The other 40 points is your poster content. Is it organized? Is it visually appealing? Does it have a logical flow, or did you kind of go all over the place? How did you analyze the data and represent it? Does it include all of those required content pieces? Then you have your presentation. Your presentation is 3 parts. There's actually you explaining your presentation. When people walk up and say, hey, tell me about your work, do you actually clearly explain the research and the data presented? That's your 30 points. And then you're going to have interaction. So as the poster presenter, you have to answer questions. So do you answer questions thoughtfully, clearly? Do you have a deep understanding of what you did and what it means? If it's really abstract, it is perfectly acceptable to be like, no, actually, I don't know. I don't have an answer for that. Let's look it up. We all have Google on our phones. Let's see what we can connect. But I'll look into that. That sort of answer is acceptable. If it's related to our research, you should hopefully be able to connect some things and pull out an answer. And then your last points about the presentation are actually as an attendee. So when you are not presenting, you are a poster attendee. 10 of your points are actually engaging with your fellow classmates and asking appropriate questions. So you must talk to each other and actually help out with their poster presentation. Finally, I'm going to leave you on this. I've talked about the Better Poster, about their research. I want you to watch at least one video, or both. Both is good. Better Poster version 1 is linked on the left. Updated research, Better Poster version 2 is on the right. You can watch either of them. I hopefully will be able to link those in the comments on my YouTube video, and also will have them linked on Brightspace. So if you have questions, please email me. I hope you have a wonderful holiday break, and we will see you next week.